Hello everyone, welcome to BioSpecificzz, your trusted space for high-quality biology education. Today, we are diving deep into the structure of carbohydrates, one of the most essential biomolecules in biology. This video will guide you through the classifications, configurations, and functions of carbohydrates in a way that is precise, structured, and academically strong. Whether you are a student, educator, or biology enthusiast, this presentation will give you clarity and depth. So to start with, the structure of carbohydrates. Carbohydrates are organic biomolecules composed primarily of carbon, hydrogen, and oxygen, generally following the empirical formula CH2ON. They may exist as monosaccharides like glucose, disaccharides such as sucrose, or complex polymers like starch and cellulose. Their biological significance cannot be overstated, serving in energy storage as in glycogen and starch, structural integrity like cellulose, and cell communication via glycoproteins and glycolipids. We will now unpack their classifications in detail. Classification of carbohydrates. Carbohydrates are classified based on the number of sugar units they have. They can be divided into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. Monosaccharides are single sugar molecules like glucose. Disaccharides are two sugar units, for example, sucrose. Oligosaccharides are short chains containing 3 to 10 sugar units, while polysaccharides are long chains of more than 10 sugar units, such as starch or cellulose. They can also be categorized functionally as aldoses containing an aldehyde group, ketoses containing a ketone group, and based on their reactivity, they can be classified as reducing or non-reducing sugars. So, if you're finding this explanation helpful, don't forget to like this video, subscribe to BioSpecificzz, and hit the notification bell so you never miss out on our upcoming lectures on biomolecules and beyond. Now coming to the monosaccharides. Monosaccharides are the simplest form of carbohydrates, incapable of being hydrolyzed further. They are the building blocks of more complex carbohydrates, and they exist in both linear and cyclic forms. Key examples include glucose, fructose, and galactose. Their chemical properties and structural variations provide the foundation for the diversity of carbohydrate functions. The D and L system refers to the configuration based on glyceraldehyde, the simplest chiral sugar. In D sugars, the hydroxyl group on the chiral carbon farthest from the carbonyl is on the right side. So most naturally occurring sugars in biological systems are in the D form. This structural detail becomes vital when studying isomers and enzyme activities or enzyme specificities. Now coming to the Haworth and the Fischer projections. The Fischer projection is a 2D linear representation of the sugar molecule, ideal for highlighting chirality, while in contrast, the Haworth projection depicts the cyclic structure more accurately, representing sugars as they exist in nature. The orientation of hydroxyl groups in Fischer forms determines whether the resulting ring is in alpha or in the beta state. Now coming to the epimers and anomers. Epimers differ at only one chiral center. A special case of epimers are anomers, which arise when a sugar cyclizes and forms a new chiral center at the monomeric carbon. The alpha anomer has the OH group trans to the CH2OH group, while in the beta anomer it is in the cis configuration. These small differences influences the molecular recognition and functions. Now coming to a very important phenomenon, that is mutarotation. Mutarotation refers to the interconversion between alpha and the beta forms or beta anomers of a sugar in aqueous solution through an open chain intermediate. For glucose, this equilibrium process usually favors the beta form, which is more stable due to reduced steric hindrance. In a mixture of alpha and beta, which is also called a racemic mixture, we lose the chirality, that is, it is unable to rotate the polarized light from right to left or left to right. While in case of the alpha and beta anomers, individually, they can rotate the plane of polarized light. If the ratio of alpha and beta is 1:1 in a particular mixture, which is called a racemic mixture, it does not have any capability to rotate the plane of polarized light. Now coming to the disaccharides. Disaccharides are formed via glycosidic bonds between two monosaccharide units in a condensation reaction. This involves the loss of a water molecule. The bond may be alpha or beta in nature, depending on the configuration at the anomeric carbon. Disaccharides like sucrose, lactose, and maltose play significant roles in nutrition and metabolism and are hydrolyzed by specific enzymes. If you're watching BioSpecificzz, if you are learning something new today, support us by sharing this video with your classmates, and for more advanced biology content, keep your eyes on this channel. Now coming to the important functions of carbohydrates. Carbohydrates are indispensable to life processes. They serve as the primary source of energy, with glucose being the universal fuel for brain and muscle function. They provide energy storage in the form of glycogen in animals and starch in plants. Structural roles are fulfilled by polysaccharides like cellulose and chitin. They are critical in recognition of cell signaling molecules and immune responses, where they function as glycoproteins and glycolipids, which act as signaling molecules. The dietary fibers, though indigestible, regulate glucose levels, support gut health, and help to lower cholesterol. Now, to quickly summarize what we have learned today. Carbohydrates range from simple monosaccharides to complex polysaccharides. Their structure determines their classification, function, and reactivity. They play vital roles in energy metabolism, structural integrity, and cell signaling. A clear understanding of these biomolecules sets the foundation for deeper learning in biochemistry, nutrition, as well as in cell biology. Thank you for watching this detailed breakdown of carbohydrate structure with BioSpecificzz. If you enjoyed this session, like this video, subscribe to our channel, and leave a comment if you have questions or suggestions for the future topics. Keep exploring the wonders of biology with us. Until next time, stay curious and keep learning. Thank you.